Hello everyone. I am grateful to the organizers for giving me an opportunity to present in this conference. I am Dr. Suchetan from Department of Chemistry at IIT Bhilai. In this presentation, I'll share our recent results on the development of hydrogel that can release insulin in a glucose responsive manner, just like a pancreas does in our body. As we all know, it takes a lot of effort and cost for one to develop a drug. However, many drugs fail to cure disease or show severe side effects due to the delivery issues. For example, anti-cancer drugs show major side effects as it also affects the normal tissue. Some drugs are not activated at proper time, such as insulin during diabetes treatment, which I am going to elaborate on this presentation. In our lab, we are interested in solving the delivery problem of drugs by attaching biocompatible polymers or biopolymers to the drug molecule. We are particularly interested in biopolymers such as protein, nucleic acid, and carbohydrates due to their biocompatibility, huge natural abundance. Further, the inherent programmability and intelligence of biopolymers may lead to improved delivery properties of drug conjugates that would help drug to be delivered to proper location and time. We also work on hydrogels to deliver anti-cancer drugs as well as antibiotic. So today we are basically going to talk about how we are using this hydrogel for smart insulin delivery. Now let me elaborate on the problem we are addressing in our lab. As we know, our blood glucose level is controlled by a closed loop insulin production from pancreas. Whenever there is a high glucose level in our body, there is a secretion of insulin. In type 2 diabetes, insulin production is diminished, leading to high blood glucose level. And in type 1 diabetes, there is no production of insulin. So, insulin injection is prescribed before the meals for type 1 and advanced type 2 diabetes patients, which of course does not mimic the natural insulin secretion. Further, it is inconvenient and painful, and it can cause ulceration and lead to fetal hypoglycemia. The existing technologies are insulin pumps and long-acting insulin, which have their own drawback. Mostly, they are too expensive to be afforded by the middle-class population where diabetes is more prevalent. To address this problem, we are developing hydrogels that can host insulin and degrade under hyperglycemic conditions only, and therefore release the embedded insulin molecule when blood glucose levels are high. We are developing hydrogels that are inexpensive and made out of biocompatible polymer and biodegradable polymers and have optimum glucose responsiveness that we'll discuss in further slides. Before we go into details of the results, we have to remember two mechanisms by which insulin may come out from the hydrogel. First is the diffusion control mechanism, in which insulin comes out from the gel irrespective of the degradation. The second mechanism is degradation control mechanism, where the drug is released via chemical degradation of the gel. The first kind of gel was prepared from cross-linking a biocompatible polymer, polyvinyl alcohol, with pinacolytic esters of bisboronic acid via trans-esterification reaction. The boronate ester bonds react with glucose molecule, which contains multiple hydroxyl group, and show degradation and release of insulin under physiological conditions. Now, the crosslinker can create interchain or intrachain crosslinks. Increased interchain crosslink would enhance the hardness of the gel as well as it will decrease the glucose-induced degradation. We postulated that by changing the nature of the crosslinker, the mechanical properties of the gel will be tuned and therefore the insulin release characteristics may be changed. We selected 4 commercially available pinacolytic esters of bisboronic acid shown here as L1, L2, L3 and L4. Although we fabricated the hydrogel under same condition, we observed drastically different rheological properties. The storage modulus decrease when we go from hydrogel 1 to hydrogel 4, indicating a hard gel is being formed in the left hand side, hydrogel 1, and other gels becomes gradually softer when you move left to right. When we studied the insulin release from the hydrogel, what we found was very interesting. We selected 3 different hydrogels with different hardness and carried out the insulin release assay under no glucose condition, that is shown in black curve, and high glucose concentration like 100 millimolar glucose concentration, that is shown in red curve. Clearly, for hard gels, there is no difference between without glucose condition and with glucose condition. Therefore, the release is primarily diffusion controlled. However, for the gels in the middle and in the right, there is a significant increase of glucose mediated insulin release in glucose rich conditions. Therefore, we conclude that softer gels with glucose responsive bonds will lead to larger insulin release. Therefore, by changing the molecular linker, one can control the mechanical properties of the hydrogel. Further, one can control the glucose responsive insulin release properties from the hydrogel. We further found that mechanical properties of the hydrogel will depend on how we make the hydrogel. For example, we can fabricate the hydrogels with polyphenol alcohol and bisboronic acid. It will easily form hydrogel, and we have selected these 3 different crosslinker for our study. So you can clearly see altered fabrication condition drastically change the rheological properties. For example, we can derive stronger hydrogel using tetrahydroxydiboron crosslinker compared to the benzene bisboronic acid crosslinker. After the fabrication, we studied the degradation of the hydrogels under various pH, glucose and hydrogen peroxide concentrations. What we found is really very interesting. We have studied the degradation properties of the hydrogel under different pH conditions, starting from pH 9 going to very low pH, pH 4. What we have seen is softer hydrogel degrades under these low pH conditions or acidic pH conditions, but the harder hydrogel don't change any weight. Similarly, when we studied glucose responsive degradation of this hydrogel under increasing glucose concentration, what we found was the softer hydrogels were degraded under high glucose concentrations, where the hard hydrogel, it was not being degraded by glucose. We further investigated the hydrogen peroxide mediated degradation of the hydrogel, where we found that the hydrogel 3, which is the hard hydrogel, was found to be degrading much more faster compared to the other 2 hydrogel like hydrogen 1 and hydrogen 2. This is because of the oxidation of boron-boron bond, which is highly oxidizable compared to the boron-carbon bond in this hydrogel 1 and hydrogel 2. So upon the exposure of hydrogen peroxide, this gel gets degraded very easily. These observations have led us to design a glucose-specific hydrogel. We used tetrahydroxydiboron containing hydrogel system that were not responsive to glucose and incorporated an enzyme called glucose oxidase. Glucose oxidase in the presence of glucose produces hydrogen peroxide in situ, and this hydrogen peroxide oxidizes boron-boron bond, and therefore it degrades the hydrogel. We observe the same under high glucose concentration. As the glucose concentration increased, the weight of the gel decreased over several hours. These results are further confirmed by visual inspection of the hydrogel. Remember, these gels do not degrade without the presence of glucose oxidase under the same glucose concentrations. This degradation of this hydrogel is very specific to glucose, as you can see in this figure. Here you have two different gel that is without any glucose oxidase and two different gel that is with glucose oxidase. When these gels are exposed in the glucose or galactose or mannose or fructose solution, only in the presence of glucose over 4 hours you can see this gel in the presence of glucose is degraded. All the other different carbohydrates does not degrade the hydrogel over time. So that shows the specificity of this hydrogel that is degraded only in the presence of glucose. We then tested the release of different molecules under no glucose and high glucose conditions, with or without glucose oxidase being present in the gel. We found that negatively charged small molecules such as fluorescein is released irrespective of the conditions. Here, however, the positively charged molecule like methylene blue or macromolecular proteins like bovine serum albumin and insulin, they are released significantly higher amount under a high glucose environment and in the presence of glucose oxidase. These results suggest diffusion control release mechanism is significantly hindered when you have a positively charged cargo or larger macromolecule inside the gel. Currently, we are developing glucose-responsive hydrogel that can release insulin out of more sustainable and abundant molecule. For example, we can take alginic acid that is derived from seaweed. We can use serum proteins out of serum, and we can also use chitosan. And we are developing different kinds of glucose responsive hydrogel derived from these sources and for better drug delivery, better insulin delivery applications. To conclude, we have developed glucose specific hydrogel which release insulin in physiologically relevant hyperglycemic environment, that is mimicking the properties of pancreas. And currently we are developing protein and carbohydrate based hydrogel for better biocompatibility. And of course we have to do in vivo experiments to find out the cytotoxicities of this hydrogel. So long-term goal of my lab is to have a pain-free oral delivery of insulin in human. Okay, with that, I want to thank my funding agencies for this project, SERB and DBT, as well as startup funding from IIT Vilai, and all my students who worked tirelessly during this pandemic time to make this project successful. And with that, I want to thank the organizers one more time, and I'll be happy to take any questions.